Hi everyone. Today I am going to discuss about polymerase chain reaction or DNA amplification. PCR stands for polymerase chain reaction. It is a DNA amplification method developed in 1984 by Carey Mullis. PCR can be defined as technique for generating large quantities of a specified DNA. It is a cell-free amplification technique. Otherwise, we can define it as the technique used to obtain unlimited supply of identical copies of a gene sequence. Next, we can discuss the principle of the PCR. The DNA of interest is denatured to separate into two individual strands. That is the denaturation step. Then each strand is allowed to hybridize with a primer. That is renaturation. Then the primer template duplex is used for DNA synthesis. That is synthesis step. The DNA synthesis is carried out by the enzyme DNA polymerase. These three steps, that is denaturation, renaturation and DNA synthesis, are repeated again and again to generate multiple forms of DNA. PCR reaction is done with the help of a thermal cycler. Thermal cycler is a microprocessor for controlled temperature cycling. In this cycle, the process is automatically repeated 20 to 30 times. So within a small time, a billion copies of particular sequence can be produced. In this process, thermostable enzymes are used. Example, Vent polymerase. Requirements of PCR that are target DNA, 2 primers, 4 deoxyribonucleotides and DNA polymerase. DNA polymerase is the enzyme used in the PCR and it is a thermostable enzyme isolated from the bacterium Thermus aquaticus. Hence the name Taq DNA polymerase. Another enzyme used for the PCR is the Klenov fragment of Escherichia coli DNA polymerase. Then the primers are used in this process. Primers are oligonucleotides of 17 to 30 nucleotides length. And the primers are complementary to the regions flanking the target DNA. It plays a significant role in determining the PCR. Primer without secondary structure and without complementary among themselves are ideal. Complementary primer can hybridize to form a primer dimer and get amplified in PCR. This primer dimer prevents the multiplication of target DNA. Next, target DNA. It is 100 to 35,000 base pairs in length. Shorter the sequence of the DNA, then better the efficiency of the PCR. Okay. The sequence of the target DNA is also important. So, the size of the DNA and the sequence of the DNA are important in the PCR. Another one is promoters and inhibitors. Addition of the protein like bovine serum albumin enhance the PCR. This enhancement is by protecting the enzyme DNA polymerase. One of the example for the inhibitor is humic acid. Humic acid found in archaeological samples of the target DNA, which can inhibit the polymerase chain reaction. PCR reaction is done with the help of a microprocessor called thermal cycler. It is used for controlled temperature cycling. The thermal cycler is programmed in such a way to provide the cycles of 94 degrees Celsius for 1 minute, 55 degrees Celsius for 1.5 minute, and 72 degrees Celsius for 1 minute in the reaction tube. PCR is carried out in a single tube kept in thermal cycler, which is programmable to set alternative cooling and heating. So the tubes contain the DNA to be amplified, reagents, 2 types of oligonucleotide primers, then deoxyribonucleotides and tag DNA polymerase. The reagents include tris HCl buffer, magnesium chloride and potassium chloride. These three act as cofactor for the enzyme reaction. Magnesium chloride and HCl and KCl act as cofactor. This reagents helps to maintain the pH between 6.8 to 7 during the thermal cycler. Primer attached to one of the strand and provide 3-OH group for polymerization. So, the DNA, reagents, primers, deoxyribonucleotides and tag DNA polymerase are added to the tube. Over this mixture, a thin film of mineral oil is poured in order to prevent the evaporation during the process in thermal cycler. This is the experimental setup of PCR. Next, we can discuss the PCR stages, that are denaturation, renaturation or annealing, and synthesis. First one, denaturation. In the first step, the reaction mixture is heated to 95 degrees Celsius for 1 minute. This temperature denatured the double-stranded DNA into 2 individual strands. That is, single-stranded DNA. Then next step, renaturation or annealing. In the second step, the reaction mixture is slowly cooled to 55 degrees Celsius for 1.5 minutes. During this time, one primer binds with the 3' end of the one strand and the other primer binds with the 3' end of the complementary strand. The process is called primer annealing or renaturation. The concentration of primer ensures the annealing between DNA and primer rather than between the 2 strands of DNA. Okay. Then the third stage, that is the main stage of the PCR, synthesis. In this step, the reaction mixture provides a temperature of 72 degrees Celsius for 1 minute. At this temperature, the enzyme Taq DNA polymerase can work properly, and this enzyme adds complementary nucleotides to the 3'OH group of the primer. As a result, the second strand grown in full size. It is called polymerase activity. This process is similar to the replication on the leading strand. The reaction can be stopped by increasing the temperature up to 95 degrees Celsius. At the end of the first cycle, two identical double-stranded DNA are formed. Each cycle completes within 5 minutes. In the normal practice, it is carried out in PCR machine. Each duplex DNA formed in the first cycle entered to the several cycles. That is, the cycle is repeated several times. The basic reaction of PCR are same in all cycles. In each cycle, the number of duplex DNAs get doubled. That is, the number changes from 2 to 4, 4 to 8, then to 16, 32, 64, etc. PCR give good result of DNA is less than 300 base pair. If we need to amplify larger DNA, then small units are amplified separately and then joined together. This picture gives an idea about PCR process. Okay. We can move to the applications of the PCR. PCR have several applications in various fields. The applications of PCR are too many to be listed here. Some of them are given here. Okay. First application, PCR provide direct information of the DNA. PCR used to detect bacterial infection. Example, tuberculosis caused by Mycobacterium tuberculosis. Another application is in the diagnosis of the cancer. Example, cervical cancer caused by human papilloma virus can be diagnosed by PCR. And also, some cancers which occur due to chromosomal translocation involving known genes are identified by PCR. Then, the PCR technique used to diagnose the retroviral infection. That is, PCR from cDNA is used to diagnose retroviral infection. Example, HIV infection. Human immunodeficiency virus infection can be diagnosed with the help of PCR. Another application is the prenatal diagnosis of inherited disease. It is by using chorionic villus samples or cells from amniocentesis. So, the diseases like sickle cell anemia, phenylketonuria, and beta thalassemia can be detected by PCR. Next application, PCR used in the study of genome. The differences in the genome of two organisms can be measured by PCR with random primers. The products are separated by electrophoresis for comparative identification. And also, PCR is important in the study of evolutionary biology. That is, we can say specifically as phylogenetic study. PCR used in the clinical field to identify various viral, bacterial, and other diseases. Another application of PCR is in the gene manipulation and expression studies. The advantage with PCR is that the primer need not have complementary sequences for the target DNA. Therefore, the sequence of the nucleotides in a piece of the gene can be manipulated and amplified by PCR. PCR is important in the study of mRNAs. mRNA means messenger RNAs. Okay. This is carried out by reverse transcription PCR. PCR used in the sex determination of embryos. Sex of the human and livestock embryos fertilized in vitro can be determined by PCR by using primers and DNA probes with specific for sex chromosomes. PCR used in the DNA sequencing method. It is a simpler and quicker to amplify the DNA. In this purpose, the single strands of DNA are required. So, these are some of the examples for the applications of PCR. I hope you all get an idea about the basic process of PCR. Thank you. Thank you all.